Yes, Karishma, we can see your slides.
Great. Okay, so they are moving. Excellent. All right. Then, Anirban, we have a little bit of an introduction um, to you, to India Bioscience, about the book chat, and then we'll dive into the questions. Also, the questions I sent you, Anirban, I have read the entire book, and the questions are just, milestones like placekeepers. We can keep it very free flowing.
Yeah, I like your questions though, because each of those is like I was thinking when I was writing them that each of those could be like a separate book of itself, especially with everything that
Okay.
happened with NDM1 and everything else. So yeah, I'm fine with your
Oh,
approach.
great. Great. Excellent. Good to know. Yeah. Then we have a fun giveaway also with a secret judge planned at the end of the book reading, the book discussion. So, yeah.
So, Anirban, we are live streaming on YouTube and we'll also be recording this session. So, if there are any questions or people are watching from YouTube, then probably I'll direct those questions to you here.
Sounds good.
So, Anirban, I see that you're on the you're editorial director at ASM.
Yes.
Yes. Oh, that's great. I have a long association with ASM, so I did my PhD in the US at UT Austin and attended several ASM conferences in New Orleans, Boston, San Francisco, and then we had a state chapter at Texas Tech Lubbock. Attended that one.
Right. What is your
So one of your,
specialization?
so I then ran clinical microbiology, but my
Okay,
research
you
group
so you
focused
are.
on
I never
biofilms.
asked, but
Yeah,
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 okay.
yes,
Yeah, yeah.
yes, yes. So this is right up my alley. This book, and one of the places you make a distinction between antibiotic and antibacterial. And that, if you know Mimi Goldberg,
Yeah,
ASM's Mimi Goldberg,
right,
she
right.
brought this up in every conference. So your book took me back in time to all those conversations. The
Yeah,
long, long association with ASM. Yeah.
yeah.
Most recently,
Even
the
now,
last conference I attended was there one in New Orleans.
next
Post
week we
then
are
I
going
haven't.
to
So
Atlanta. Next week our ASM
exactly,
microbe is going
exactly.
to. It's
Yes,
like now
yes.
they've combined both ECAC and the ASM meeting, and now it's like called
Okay, I
ASM
see. I
microbe.
see.
So it
I
will be
see.
in Atlanta
Okay.
next week.
I have also attended an ASM conference for undergraduate educators, the ASM CUE.
ASM CUE. Yes, 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 yes.
Yeah, yeah. That also I have attended. Lots of I received a fellowship to attend it actually when I was faculty at UT Austin. So really great initiatives by ASM. And I saw that you're advertising a position in India.
Yes, we are actually, we got a lot of responses through the IndiaBioscience.org ad. And we've actually started to interview people. So I'm really impressed
Excellent,
with the candidates that we
excellent.
got through
Oh,
that ad.
excellent.
Thank
So
you.
post this, Anirban, I will be in touch on email with a proposition related to that. I have
Okay,
some ideas related to that. So
great,
I'll
great.
be in touch. Yeah, it's really good to see ASM have a footprint in India.
Oh,
Really great.
absolutely.
I mean,
We
it's
needed to
always
do
had it through
that.
the ambassador program. An ambassador program has always been there, but nothing more than that.
Yeah,
So
and it's
really
not
great,
adequate
really
for the
great.
country of the size of India and the
Uh,
like the needs there.
Precisely,
So yeah,
precisely,
yeah.
precisely, exactly, exactly. So
Karishma,
really good to see. And I have a proposition which I'll get back to you on. Yes, I'm not trying to make a hard sell.
No, 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 no. Yeah, no, absolutely. Yeah, I understand. We're looking
Just
for these opportunities. And yeah,
Karishma, just
Great,
sorry to
Anirban.
interrupt you, but I just wanted to let you know that if you could keep your screen on full screen, currently your tabs,
It is,
everything visible.
it's, I don't know, how do I do that? It is, what do
This
I
is
do
on
now
Google
to
Slides.
that?
Is this on
It
Google
is through the
Slides?
drive, that's right.
Yeah, but if it's slides, then you can probably make it full screen, like put it in slideshow mode. If this is a PDF, then probably it will,
No,
uh,
it's not a PDF, Harushi. It's through, okay, see, let me see. Let me stop share.
yeah.
Okay. And I will reshare and see, I'm here.
Yeah,
Now,
can you click on
what do I do?
slideshow?
Even if I click on slideshow, which is what I did last time, the tab, acha, now it's showing up like this. Okay, okay, thank you. Maybe I did something different. Correct. Perfect. Thank you. Now, can you see my screen?
It is still loading.
That's the thing. The moment I do this, it stops sharing.
Okay, then I think the previous one was fine then.
Okay.
Yeah, or maybe if you can download it as a PDF and then,
Yeah. Yeah, sure. Let me see if that works. In the meantime, we can get people into the meeting
yeah,
room.
I'll admit them.
Yeah, we need to have a separate conversation. It's really nice to meet you.
Yes, definitely, Anirban. We'll do that. My silence. Okay. I guess now it's all visible, right? Excellent. All right. I think we will slowly but surely get started. Um, yeah, just a second. I'll just keep this here for a few minutes till I finish the introduction, and then I'll move into the full screen share mode because I just have some notes that I need to refer to. All right, so I think it's 10 o'clock. Welcome everybody. Anirban, it's way past midnight, you said at your end. So bugs and drugs keep us awake, right? Let's just start with that. They give us sleepless nights for good reasons, not so good reasons. We'll talk about that. We have a lot to talk about. And right here, if you can see, is the book. Is the book we are going to be discussing, When the Drugs Don't Work, the Hidden Pandemic That Could End Modern Medicine, with the author Anirban Mahapatra. So what a wonderful opportunity this is to tell you a little bit about Anirban. Anirban wears many hats, and that's something, wears many professional hats, and that's something we're very familiar with at India Bioscience. So Anirban is a scientist, an author, and the editorial director at the American Society for Microbiology. He's passionate about bridging the gap between scientific research that happens in our universities and higher institutes and public awareness. And he does so through writing, through conversations, talks, and other professional work. One of the examples of him attempting to bridge this gap is this book, When the Drugs Don't Work. And it's his second book, the first one being on COVID-19. So India Bioscience would thank you, Anirban, right at the beginning for being a part of this, for coming on board so quickly. And thanks Juggernaut Books for this wonderful partnership, which includes not only this virtual book chat, but also a giveaway. So for those of you who are joining us, you have plenty of incentive to stay tuned and keep it active through the chat window, because thanks to Juggernaut Books, we have a virtual giveaway, and there is a secret judge who is one among us, revealed, that will be revealed towards the end of the chat. But to get a free copy of this book shipped to right at your doorstep from India Bioscience, you will need to keep the chat window active and ask really good, thought-provoking questions. So how does that sound? Sounds like a good way to start Friday. Yeah, a good way to embark on the weekend. Maybe we have some thumbs up in the chat window for those of us ready to start the weekend. Of course, after this book chat. So some thumbs up. Very good to see people joining in. Thank you so much. Good to see you all. We expect this to be a very focused and engaging discussion. All right. Great Friday, says Ashwini. Thank you, Ashwini. Okay, so moving on, a little bit about India Bioscience. So we are a science facilitation organization based in Bangalore, but we work across India. We are largely funded by the Department of Biotechnology. We have multiple external partnerships as well. So we work with stakeholders who are academia, industry, government bodies, not-for-profit entities, broadly related to the life sciences. And we serve as a catalyst, a facilitator, engagement partner to spark conversations, collaborations, and ideas. So we are a small team. I will introduce the team shortly, but we work across these many verticals, as you can see. So networking, skill building, education, community building, communication, digital initiatives, and data and policy. And all of these initiatives feed into our platforms. Our website is our one-stop place to get information, what's going on at India Bioscience, and we update our website multiple times a day. Yes. All right. Our other digital platforms are a webinar platform, podcast platform, e-com media through India Bio Reads, and we also have a new series on short snippets, which you largely find on Instagram and social media, which is India Bio Snippets. In addition to this, our team of 7 people travel and work across India for in-person workshops, events, meetings, and I'm sure you will find us somewhere and come by and say hi. So this virtual book chat supported by Juggernaut Books is part of India Bio Reads. In addition to book chats, India Bio Reads publishes e-compendia across the year. So you can keep an eye out for our many publications, the most recent one being in the month of May. So lots happening at India Bio Reads, lots of content for you to enjoy. And all of this, believe it or not, gets done by a team of 7 people, and that's who we are. All right. So if you meet us at an in-person event, come by and say hi. Of course, if you see us at a webinar, our chat windows are open. So with that, I'm going to welcome you, Anirban, once again, and we're really looking forward to this discussion. I think at this point, should I stop sharing my slides and just go into the questions, Arushi? What do you think? Yeah, because it's the same slide. Okay.
Yes.
So maybe then you can, thank you, Arushi. You can pin Anirban. Okay, so Anirban, lots to talk about your amazing book. Really, I dug into it. Dug being a pun for prospecting antimicrobials from soil. So there are going to be a lot of puns. Some you will understand, some you will not. Never mind. Just let's go along with the flow. Okay, so let's start right at the beginning, Anirban. In recent times, India woke up to antimicrobial resistance in a very big way, and probably one of the most, I would say, it captured public attention very strongly, which was the New Delhi metallo-beta lactamase story. So there was a lot to be discussed there. What was this bug? Why did it have this potential to spread so rapidly, not only in South Asia, but there were also reports of identification of NDM in Europe. So maybe you can tell us a little bit about your research in that. It's covered extensively in the book. And finally, maybe you touch upon this fact that there was also a lot of controversy surrounding the nomenclature, calling it the New Delhi strain. And more recently, there was a lot of propaganda to not call COVID the Wuhan strain, and so on. So you also
Yeah.
do touch upon that in your book, that did India not object strongly enough to it being named the New Delhi strain? Or is this standard nomenclature when it comes to microbial strains? We want to hear it from you. But to start with, you tell us a little bit about NDM and the controversy it stirred up in India.
Yeah, absolutely. So first of all, thank you, Karishma, for inviting me and for the praise of my book. And I'm really looking forward to the interactions today and discussions after this as well. As most of you know, like there's so many issues with respect to science and bioscience that impact us globally, but also in India. So I've maintained my ties. But writing a book is a very lonely act, especially writing in a topic that isn't such as if you're not writing about politics or films or cricket or any one of those topics that are very, very sort of very popular right now. So writing one book, popular science book, well, at least COVID was at that time we were in a pandemic, but moving on to that and writing a second book. So it's really great to speak in front of my tribe, people who are interested in this and are moving forward and are champions of both popular science, science popularization, but also the need for scientific discovery. So this book, it's the original sort of seed of this book came from at least around the time of the New Delhi metallo-beta-lactamase saga or NDM1 saga that happened in 2009 and then 2010. At that point, like it was in the media, and somebody told me at that time, you should like because I was chiming in on social media. I had just joined Twitter, which is now X, and was commenting on that from the perspective of being a microbiologist. And somebody said you should write about this book because this is something that like when we talk about science, like we talk about it from taking it outside from the lab and from what we understand to how it affects society broadly. And that was a case where stuff that we were doing or we were discussing in labs or that we had studied for, let's say, our PhD or other work, moved forward and moved into a very high level. So first, before I get started, I want to say that thank you again for the introduction. Everything that I'll say here is my own personal opinion. It has nothing to do with my organization ASM or with the journals that I work on or any of the other people represented there. So moving back to what happened at that time with NDM1. So you may even recall like even was debated in Parliament, which doesn't very often happen for something that is scientifically relevant. So at that point, I thought, well, okay, so this is something we need to trace like what's happening here. But as happens in most things, like it makes like the news cycle is about 24 hours. So it makes the news, it's either a headline, it's a sound bite, somebody says something, and then it's gone. And then people forgot about it. And then even now you'll see like superbugs come out in sort of either a very sort of scary or dire way in that, okay, this is like a superbug, this is happening. And like they go to some hospital, then they find like all of these patients, and then it's like just covered there, or it's something else that is like somebody in a lab has made like this discovery, and it's like okay, it's like they found an antibiotic that will like defeat superbugs, and neither of those is the full picture. So at that point, like when NDM came up and then these other subsequent events, I thought this is something that needs like a deeper coverage. And at that point, I was thinking like maybe I should write a book. Well, in between, COVID-19 happened, and so I wrote a different book and then came back to this because of some personal stories that people had been telling me. So if you the NDM1 story is very interesting because what happened in 2009, it's almost like a medical mystery, which is what we see a lot in microbiology. In 2009, a patient who lived in Sweden, he came to India and he visited his family in Punjab. And then he was treated for an infection in Delhi, New Delhi, at a clinic. And it was an infection that just wouldn't get resolved. Like they gave him a variety of antibiotics and he just wouldn't get better. So once he went back to Sweden, the clinical microbiologists there did a whole battery of analyses, and they found that he had a persistent infection, a couple of persistent infections in fact. And then when they tried to identify it, they sequenced it. They found that it was a completely new resistance gene that they had found at that point. And so they published it in 2009 in a journal. And at that point, that's when it was named the New Delhi metallo-beta-lactamase 1 or NDM1. Now, this was keeping in convention with what had been done in the past. Like there are resistance genes named after Verona, São Paulo in Brazil, Guyana as well. So they didn't give much thought to that. That was one of the microbiological conventions. And that paper actually, that 2009 paper didn't make any waves. It was not covered in the media. People forgot about it because it was just one patient, right? What was happening, nobody really cared, apart from the microbiologists. And then in 2010, what happened is then a new journal at that time, Lancet Infectious Diseases, they published a report that said like this was now found not just in India, but Pakistan and Bangladesh. And they did a few examinations and tests, and they found that it was more prevalent. And at that point, they also made some rather controversial recommendations in the discussion of that paper, where they said that this can be a huge threat, and people from Western countries should not go to India for medical tourism or surgical operations at that were coming up at that time. And this caused a huge furor. Just to give you some of the statistics, we are lucky if our articles get cited even a few times as researchers.
You
This
said it.
this
You said
article
it.
since then has been cited about 2,000 times, and even in the first 4 or 5 days, it was downloaded about 5 million times, and people were reading it who had no knowledge of microbiology, like what was going on there. So
You mean the Lancet Infectious
that
Diseases article?
Lancet Infectious Diseases paper, like 5 million
Okay.
downloads within a couple of days. And it became one of the Lancet Infectious Diseases, one of the most cited articles in that journal's history since then. And I mentioned what the most cited is, and maybe we'll talk about that one as well. But this was the second most cited. And the reason for so much concern that the scientists put at that time is because the resistance was to a class of antibiotics called carbapenems. And they were at that time, they were the state of the art. Like if you're giving antibiotics and if susceptibility tests are not done, then the doctor may move on from one antibiotic to the next and see how well how well your infection is responding. But at the carbapenems were thought of as antibiotics of last resort. So if antibiotics don't work and you have a resistance gene at that point, then the treatment options are very, very limited. The other problem was that it was found on a plasmid, so it was very easily transferable through this process known as horizontal gene transfer, which whereby the resistance can pass from one form of a bacteria to another. And that's what it was observed. Like it started off in a couple of bugs such as Klebsiella and E. coli, but then it was spreading among a lot of gram negatives. And this was a cause of concern. And so the researchers went back and 2011, they published a follow-up paper. And in that paper, they found what they had feared. And that was that resistance gene was now found in unrelated bacteria from water samples. So it had now been found in the environment, in water samples collected across various parts of New Delhi and northern India. And from that time, from that time, it's been seen is now that spread to, like, as you mentioned at the start, like it's been spread to many countries. That NDM1 has now like many, many variations. And I think a 2023 study just last year found that Klebsiella pneumoniae, many of them have developed resistance to carbapenems, not just through NDM1. So we want to make that clear. and that is a cause of a large number of newborn infections and like neonatal sepsis. So newborns who are in NICUs, they are passing away from this. I guess the
So,
other
Anirban,
question
just
you
to
asked was,
interrupt, a couple of
yeah,
points to just highlight here. One is you made a
yeah,
point that this nomenclature, whether it has, like plasmids have been named or mobile elements have been named after Verona, which interestingly is where the Shakespearean play Romeo and Juliet are based. So we could now have a modification, Romeo and Juliet and antimicrobial resistance. That would be a nice theatrical description of AMR. So it was not with any vendetta. There was no political machination against naming it after New Delhi. It was standard practice. However, because the latter part of the paper, and in this chapter, you do talk about how a paper is logically arranged from an abstract introduction to a discussion. In the latter part, they allude to the implications, which any scientist in the discussion section of the paper will include. The implications will be where they talk of medical tourism in India, elective surgeries in India, and so on. So, somewhere between the nomenclature and this very broad implication, which could affect medical economy in India, the controversy focused more on the name than everything else to do with this strain, right? And somewhere, though this strain emerged and it could have been actually used as an opportunity to really take stock of antimicrobial resistance in India, which in the book you do talk about how subsequently a study led by K. Vijayaraghavan
yeah,
looked at that very closely. And interestingly, Professor K. Vijayaraghavan is a advocate of India Bioscience. So I'm just going to put it out there. But during his tenure in the office of PSA, this study happened and we'll talk about it. But if we shift from NDM to a more sort of a compelling story, which was MCR in pigs in China, what NDM could not do, MCR did, where it actually managed to change practices in the poultry industry in China. So can you tell us a little bit about MCR? I believe it's a colistin resistance,
yes.
colistin being widely used in poultry to promote growth, and to sort of fatten up the meat. But tell us a little bit about MCR and how did it manage to do what NDM could not do? Was it because there was no controversy around the nomenclature? This
Yes, partly.
national umbrage, partly that. And tell us a little about that and how it actually managed to change poultry farming in
Yeah,
China.
great point, great segue. And it's like almost the next chapter after NDM1 is like, so when
Yes.
NDM1 was found, there was still like one antibiotic that physicians could refer to, and that's colistin, which was discovered in the 50s group, but it was not used very often because it's a highly toxic antibiotic. You only want to use it when there are no other antibiotics available. So it was an antibiotic of last resort. And interestingly, one of the authors who was actually on the NDM1 paper was an author on the mobile colistin resistance paper. So this is another resistance gene. It was not named after China or any other part of China, even though it was discovered in China. And I think he realized, and I think uh, a lot of people who I've spoken to also believe that the NDM1 gene was unfairly named. And I am one of the people who think at the time, even though there were no ulterior motives, the nomenclature should have been different as it was for the mobile colistin resistance 1 gene. You don't want to call out any place or stigmatize any place. And I don't think that was an unfortunate fallout of that entire episode, even though that was not the intention. So with the mobile colistin-1 gene, which they found, and that was discovered more recently. So that was 2015, 2016. And it actually came out over from what they think is it moved over from pigs. And the reason that happened was because colistin had not been used for many, many years. It was used in agriculture. So what a fact that most people do not know is that about 70% of antibiotics are used not for human health, but to fatten up poultry, pigs, and other livestock. Because there's another discovery that was made that I point out in my book, that instead of keeping animals in a hygienic condition and letting them grow, in that sense, a lot of antibiotics are used as growth promoters and fed to animals. But at that point, it was not recognized by the agricultural community globally and in various parts of the world that this was a critical antibiotic for human use. And as we know for COVID and bird flu and everything, there's a One Health approach in which the environment, animals, and humans are connected. So resistance does not just develop in an organism and just stay there. So it can happen, like resistance happens in pigs, it can transfer to humans and vice versa, from humans to other animals as well, especially in close proximity. So that's what was discovered. This gene for this last resort antibiotic. So you can imagine the situation here where if somebody has both the NDM1 gene and the MCR1 gene, the
MCR.
colistin gene, then there is virtually no antibiotic that will work for the type of superbug infection they have. So this is a catastrophic situation. And at that point,
Yes.
what China did was first they banned colistin for the use in agriculture. But at that point in India, it was still available, like 2018, 2019. And a couple of journalists did some investigative reporting, and they found that you could go to Venky's, and Venky's was actually selling this. You could buy it over the counter, like colistin. They were able to purchase 200 grams of this. And this has actually come up in the media reports very, very recently as well, as it was being fed to chickens. But once this was raised, the Indian government took very decisive action, and now colistin has been banned. So it's only used for human use. And I would argue that that has to be the case for all antibiotics that are critical for human health. Is we should reserve them only for human use and not use them for agricultural practices. But yeah, I think that was the situation there was very, very different, partly because of the naming of that, which I think the naming
Right.
was more appropriate.
Okay, so Anirban, questions are coming in, and we will take a few questions before we move into the sort of planned, structured discussion. Raksha asks a question. A lot of work is happening in AMR worldwide, and it's great work. But how do we integrate this to address AMR? So there's clinical work, there's scientific work, there's sequencing, there's social work, there's economic policy, public policy. But is it actually translating to anything on the ground, into integrating into effective solutions on the ground? And how could this be done better?
This is a very complex and important question, and it starts from very basic steps, such as even just basic hand washing and improving hygiene. These are things that in a clinical setting, they will reduce and separating patients and making sure that patients wear those clinical tests, clinical isolates, identify someone with resistant pathogens, that they are kept separated from others. So I think it starts with very basic practices like that. I think the second is raising awareness of this issue amongst not just physicians, but also amongst a large number of people, that you just don't take antibiotics willy-nilly just because they're available. They're harmful for your health, but they're also harmful for the broader ecosystem and for the development of resistant pathogens. So I would say that would be the second point. Third is just improving overall health and preventative practices would be very, very useful. And that includes vaccinations. And that's because if you don't get sick, then you won't get even wrongly prescribed an antibiotic. So even reducing the burden of people who are sick, I think is very, very important. And then antibiotic stewardship, which is reserving the antibiotics of last resort for truly important cases where they're necessary. So I think those are some of those things that can be done. And then finally, I would add that we need better clinical and diagnostic approaches, because I understand why people are being given antibiotics. If a confirmatory test takes 2 days, and that's the gold standard test, and there is no rapid test to identify the specific organism, the causative pathogen, you don't know if it's a virus or if it's a bacteria, and what kind of bacteria and what it's resistant to, then the doctor will say, I'm not going to see this patient again. let me just stay on the safe side and prescribe antibiotics. But if there were a cheap diagnostic test, a rapid diagnostic test with good specificity and sensitivity, then that could be a
Certainly.
more tailored treatment option. So I think all of these things are, I know it's just
Right.
scratching the
And
surface, but all of these things will help.
absolutely. Your book, of course, is much more than the surface. It really digs deep. But the question is also sort of everything that you discussed is written in that study, which was sort of the forward of that study is written by Professor K. Vijay Raghavan.
Right.
It openly states, and to come from a government source, that India has the highest some of the highest antibiotic resistance rates in the world, especially to the 6 deadly bugs, the E. coli, Pseudomonas, Klebsiella, and a few others, and also some of the highest rates of antibiotic sales without prescriptions. So it's sort of this, it's really between the devil and the deep blue sea kind of situation. And I thought it was a very forthright report with government validation. And I think that our listeners can go into that in a little more detail. But then there's another great question that comes from Ashwini, which actually also segues into what we discussed for the structure, which is there is so much of a fall through between a candidate and an actual drug in the market, right? So many of these studies that you mentioned at the beginning, they say, oh, scientists in this institute have discovered this molecule and so on. But we 5 years later see this as a packaged drug in the market? We don't. And one of the things I wanted to talk to you about is that what is the economics of antibiotic discovery and why are antibiotics a market failure? You have a whole chapter talking of why they are a market failure. But why don't pharma companies want to invest in antibiotic discovery and manufacturing when it is such a huge problem?
It is a very complex question. I think, first of all, it's become much harder to do that. During the golden age of antibiotic discovery, which happened, I think, not just during Fleming's time, because when Fleming discovered penicillin, it took a few years for the chemist to figure out actually how to make that into a drug. And I talk about that in the book as well. But he didn't patent it, and that was one of the things that led to penicillin being widely available. But then Waxman obviously and his team found streptomycin. And so if you go to and I have this written in the book as well, most of the isolates of antibiotics come from soil bacteria, especially from the actinomycetes group. So if you try to screen 10,000 actinomycetes, you will find about 2,500 of them are producing some form of antibiotic as a secondary metabolite. It's not something that they absolutely need for their growth.
And that is a 25% chance of finding an antibiotic molecule,
right,
which is not trivial.
Right. But then you go down and you see, okay, so so it's like 120. So now we're starting from that 10,000, only 125 will make streptomycin. Okay, so now the next level of that is even rarer, vancomycin. You won't even find it out of those 20,000. Vancomycin was isolated from 1 out of 100,000, so 1 out of 1 lakh. And the most recent the last innovative antibiotic, which I mentioned in my book, daptomycin, it was discovered in 1984. There were some toxicity issues, and then it became one of the last innovative antibiotics. So we haven't had innovative antibiotics in decades. That is like you have to screen at least 1 crore isolates to find that. It is such like a proverbial needle in a haystack. So it's become much harder, first of all, to find these antibiotics from their natural soil habitats. Second is we haven't moved so much out from soil into other, I mentioned this in the book as well. So uncultured organisms, oceans, and other ecosystems where there could be antibiotics. So the heartening news is even today, there's a paper in Cell that just came out by Cesar de la Fuente and his group. And what they did is they used AI to scan metagenomes, and they found some candidate antibiotics. So again, this is very early stage. It's like most of these won't even make it down the funnel where, right, you start off with a lot of candidates, then they go through various stages of preclinical toxicity, preclinical, clinical testing, and then it goes through that process. But the number of people who are actually working on antibiotics is fewer than in other fields. So I just have the statistic, this is the latest statistics. Right now, there are only 27 drug candidates in clinical development for priority bacteria. This is in comparison with 1,600 cancer treatments. So that gives you a sense of how antimicrobial resistance has been deprioritized. It's estimated that there are about 3,000 active researchers worldwide who work on this, who work on antimicrobial resistance, compared to close to 45 to 46,000 people who work on cancer research. And this is despite the fact that within another 10 or 15 years, more people will die from antimicrobial resistance than are dying right now from cancer. So it is a serious problem. And there are a number of reasons, including the market failure aspect.
Anyway, the question is sort of like, even if we find a molecule, a few molecules, a handful of them, why aren't pharma companies jumping the bandwagon to manufacture at scale?
Yeah.
So how does it just not make good commercial sense? When
it
we
doesn't
say at the
make
same
good
time,
commercial
crores of
sense
people
for
will.
them. Yeah.
Why
And
so?
the reason is it's not like the big drug right now. Everyone is talking about like, okay, Ozempic. It's like semaglutide,
Yes.
Ozempic and Wegovy. And these are the GLP-1 drugs for obesity, and they'll reduce your blood pressure. And nobody it's like it's even made it into like the influencers on Instagram are talking about that.
Yes.
And the reason it's such a big cash cow is you have to take it for your entire life. If you come off that drug, you will, short of making lifestyle changes, you will regain your weight. This is same with statins for cholesterol. So pharmaceutical companies want to invest in drugs where they have a steady stream of cash. For these drugs, let's say for an antibiotic, let's say they have all these drug candidates, it takes 20 years to develop the drug. They will have the drug, let's say maybe for 10 years, after which time it will go off patent. It will become a generic drug. So they have to recoup all their R&D costs within those 10 years. And we are just talking about like save the most advanced drugs for the only last resort cases. So how can they do that? This is like they've invested so much in this drug, and now we're telling them that you can't use it in most cases unless somebody is resistant. So that's why there's
multiple other drugs. Exactly. And also antibiotics come with a dosage regimen. You'll give it for 7 days and say stop taking it. But Ozempic is for life if you want to stay thin. And apart from the fact that an Ozempic caters to vanity.
Yes, absolutely. And it's also the other part of like who is affected by this. It is the people who are who have other infections, who are the elderly, who are hospitalized, who are infants, and they don't have the loudest voices or the fattest wallets. So it becomes an
and
issue
it's not
of
fancy
equity.
also to tout daptomycin. It's probably more fancy to tout Ozempic. But coming to that, but there are antibiotics that we have to give for years. For example, for tuberculosis, the treatment is 6 months at least. Leprosy is another example. So these chronic intracellular pathogens, why aren't people investing in making drugs for Kala Azar and tuberculosis? There is a huge market in India, South Asia, Southeast Asia, Sub-Saharan Africa. People need to take it for at least 6 months to a year. Why do they fall off the economic market value?
Yeah, this is where I will say that India has to step up, and this is where we can take. We have it costs us a lot less to go through. So the number I quoted, it's a Western number. It costs about $1 billion for a Western multinational pharmaceutical company to go through the whole process of developing a drug, but and going through the clinical trials. But I think a drug made in India, even the clinical trials will be less expensive to be able to do that. And I think the drug priorities for the diseases that you mentioned, and even moving beyond that to tropical diseases and some neglected tropical diseases as well, the priority has to come from those nations and those countries. So I don't think other countries are prioritizing it. And the large pharmaceutical companies are not. Again, it's driven by the profit motive of how much profit they can derive. So I
And
think
also, all said and done, I know Dr. Peter Hotez is a
yes,
advocate for neglected tropical diseases. Yes, I've met him at some ASM meetings, but also for someone working at CDC or NIH. Kala Azar is an esoteric tropical
right,
thing that happens to some people in some other part of the world, and it's not our problem, we don't deal with it, kind of situation. So really,
yeah,
it's correct. Neglected tropical diseases in India, Southeast Asia have to step up and do our own sort of prospecting,
right.
discovery, research, taking to market. And I think this answers Akshata's questions, coming back to the chat window, Anirban, where what's the role of the pharma industry? Great question, Akshita. It's been answered. And also, yes, antibiotic resistance happens through horizontal. In one mechanism it happens is horizontal gene transfer. And so, how exactly do antibiotics in the animal industry impact humans? How exactly does this transfer occur?
So where there is this is a trickier one. So this is basically like we know I'm going to give an analogy. We know that there's an environmental reservoir of pathogens that have these resistance genes and how they spread. And there have been environmental studies that have shown that people who are in proximity to farm animals can pick up those precise resistance genes and also pass them on. So there isn't that sort of direct link of like this person got it from this animal, but there is a link from that environmental chain that they found that. And there have been multiple studies. I think in my book, I cite like there are over 150 studies that have shown this. But the link is kind of it's an indirect link, the same way that a climate change link is like, okay, CO2 is linked to climate change. So that's the sort of link that there is. But again, it's part
Yes,
of
yes.
the same organisms are infecting multiple the same pathogens are infecting multiple species, and the same plasmids are transferred through horizontal gene transfer across species as well.
Exactly. And oftentimes, when the link gets established, it leads to this large scale culling of poultry, pigs,
Right,
and so on, which is huge economic loss apart from also environmental loss.
right.
This large scale culling. What the real root of the problem is the farming, the animal farming, poultry farming, pig farming, fish farming that has become at such a
Yeah.
scale that it is unsustainable. And to
No, I
meet that
agree.
scale, we are feeding them with these growth promoters, and basically
Yeah.
antibiotics in poultry are growth promoters.
Yeah,
And
exactly.
that's what the farming practices are doing. So I think the root problem is to reduce the farming at that scale and bring it to more smaller establishments. Okay, well, our audience seems to be giving us the questions, Anirban, that we had structured. So, and I think we also answered this great question about, Waxman's experiments, Selman Waxman's
Yeah.
experiments with soil. And for all the young scientists who are here, PhD researchers and postdocs, if there is such a plethora of microbes in this microbial, antimicrobial chemicals or compounds in the soil, uh, why aren't we all just prospecting soil? I mean, you gave us some numbers that it takes 10,000 to get
Yeah.
125 and so on, but it's largely an untapped
It's largely
cosmos,
untapped.
in alternative universe, you might call it. So, what is the hindrance to everyone doing soil experiments and trying to find actinomyces and streptomyces that are making these chemicals?
I don't think there is. I just think it's just become in many parts of the world, it has become unglamorous to do that. But this is something that I'm actually thinking that like my son is 12 years old and he has to do a science fair project. And I think like he should go out and try to do this as well, even if he isolates something. It's like basic microbiology techniques to be able to do certain things like this. And I know that there are professors, Joe Handelsman, who was a former ASM president. and she's actually run a whole program where people are high school students and others are putting together, pulling soil samples and trying to test them out for this. But I think it's a great suggestion. More people should attempt this, and I think we will discover more antibiotics that way.
I think the good way to put it is we are so obsessed or so consumed with exploring AI and what we cannot see that we are not looking at the dirt beneath our feet, which actually might be the solution, present some solutions actually. So we really need to look closer home, pun intended, right? The soil in our backyard. And then Anirban, Chetan has a great question, which also brings us to the latter part of your book, where you say beyond antibiotics. And here you touch upon vaccines. For example, if we vaccinate for rotavirus, then diarrhea in children will not wrongly as well. We will not wrongly need to prescribe antibiotics, right? You also very interestingly talk of antimicrobial peptides, of course, and one topic that we can sit with a bit, which is something that Chetan has brought up, which is phages, bacteriophages. And this is something that it sort of now phage therapy is restricted to Eastern Europe, Georgia, the former USSR states. That's sort of the only countries in the world that seem to be taking it very seriously. But your book has a large segment on phages. What, in line with what Chetan is asking, can we think of phage therapy plus antibiotic therapy as the future of this deadlock?
I think in certain cases you have to. The only issues around that is obviously it's a very tailored solution where you have to go find the specific virus that attacks that bacteria. And what happens is like this, so it's hard to do clinical trials on this, right? It becomes a very compassionate case study where somebody who's and it becomes a very compelling story, and it becomes like one case study that gets published and somebody writes a book or something like that. But at the scale of the problem, it cannot be a sole solution. I think that's one of the main issues. But I think in that case, when you have somebody, let's say, who is a loved one, and they are at that situation where now no antibiotic is working against them, why not go for, like, we are doing this for cancer. We're doing personalized therapies for cancer. So why can't we have personalized viruses in those cases for people? So I agree that this has to be one of the arms of how we treat in the future
infections, and especially persistent, recurrent, multi-drug resistant infections. And yes, phage therapy is likely to be personalized. It's likely to be like end case scenarios, which is actually Stratby's book when she talks of how she's saved her husband from a near fatal infection. And it was phages that changed that. All right, that's very interesting to think of.
She had a really like it worked out brilliantly for her. It was like she was an infectious diseases specialist, so she knew how to contact people. But even in cases where it looks like it's getting better, we have to remember that the bacteria can also develop resistance after a while. So that's what happens. Like the case sometimes gets better and then it gets worse. So because there's always like with the same thing that happens with an antibiotic. The thing with an antibiotic is that it works fast. So when it works, often it works very, very quickly. But with any of these type of biologics, it can take time. And so the bacteria can develop resistance over time as well.
To the phages as well, because finally, even between the bacteriophage, which is a virus that infects the bacteria, there is an evolutionary arms race, right? Not all phages are able to infect bacterium, but bacterium can also evolve, change their receptors, which the phage uses to enter, and so on. So again, you'll have to try a trial and error, and it'll have to be a really end case, compassionate use, no solutions available, and then you take this on. We'll take few more questions, Anirban. Yes, Chetna has another very good, apologies for mispronouncing your name, Chetna. Chetna has a very good question on, see, when we say antibiotic resistance, almost default, we say it's bacterial. But in the end of your book, you also talk about fungal resistance, parasitic resistance, which are even lesser addressed than bacterial resistance. Maybe you want to elaborate a little bit on that, Anirban, as to why you didn't include it in this book, and maybe tell us if there is another book happening on fungal and parasitic resistance.
Yeah, well, we'll see. I purposely thought like, how will I tackle this topic? So first of all, throughout the entire book, I have never used. I tried to avoid using acronyms. I don't even use AMR. Like I mentioned antimicrobial resistance, but because once you make it for a popular audience, and then I said, okay, I'm going to talk about like 6 specific organisms mainly. I mentioned TB up front, but then I tried to avoid it. I'm going to talk about these organisms which are responsible for 80% of the deaths that are predicted and the infections that happen, and all of them are bacteria. But definitely, fungi is that's a whole other issue with all of the new infections that are happening, and especially with climate change as well. Like there are so many more fungal with Candida auris and everything now showing up in clinical settings as well. So that is definitely an issue. But I think wherever I have a line in there that basically says like Leslie Orgel, the British chemist, Nobel Prize winner, said that evolution is smarter than you. So every single time when we come up against an organism, there's going to be some resistance that forms. And that's even the case with cancer. Like even with cancer drugs, with chemotherapeutics, your own cells can develop resistance against these chemotherapeutics. So that's just
Yes.
the inevitability of evolution. So to your point about if there will be a book about fungi, perhaps, like I guess that's the natural order of progression. If my first book was on like viruses, then I've gone to like unicellular organisms. The
Okay.
next one probably should be some sort of
Fungi and
multicellular.
parasite. Keep us in
Yeah,
the loop. Tip us off
absolutely.
early on. Okay. All right. Another interesting question, Anirban from Suresh from Ethiopia. Great question. One that you actually have not addressed in this book. What is your opinion on the effectiveness of herbal formulations for AMR?
Yeah, so herbal formulations at a point before the introduction of antibiotics were common, and there is some evidence that some herbal compounds have some antibacterial germicidal activities as well. So I would have to say that we should definitely look at them and see what the natural product is, what the active compound is, but it should, I think, whatever the compound is, there should be some medical backing through trials, definitely.
Certainly, and not herbal concoctions. Like
Yes,
potions and things that we really
nothing
don't
like
know
that.
what the
Yeah.
active ingredient is and so on. Exactly. Okay. Ashwini has a great question on the future. So coming from herbal formulations like poultices and potions, which were ancient remedies, we are going all the way into the future. I mean, we are spanning time in this 1 hour webinar, Anirban. But Ashwini's question is, what's the future of AI in antibiotic discovery? Or are we going to stick to Waxman's experiments with soil? Which
No,
could
actually,
be.
there are already a number of experiments. I mentioned a few that are in my book. One of the groups that is working on this, I think somebody else might have also put it in the chat, is like Jim Collins' group at MIT has been using AI to try to. I mentioned the 6 deadly bugs. They are going through each one of those and trying to identify chemically synthesized compounds through that approach. So they're basically what they're doing is they use a machine learning algorithm to look at which antibiotics work already, and they are now using that algorithm to test it against the entire chemical space of created compounds. And they had one paper, I think, in Cell, one paper in maybe in one of the Nature, I think two in the Nature journals for various organisms. Then there was the paper I mentioned that just came out in Cell, Cesar de la Fuentes' group. And what he did was basically use AI for not for chemically synthesized compounds, but used it as an approach for screening. So basically took all of the metagenomic data and said, okay, this is what an antibiotic looks like from the genetic data, and try to identify the genes, the cryptic genes for antibiotics, Then he used those to synthesize about 100 of them and found some antiviral. So what AI is doing is actually speeding up that early discovery process so that we can look at more compounds and we can sort them faster without spending that much money. I think another aspect of where AI can be used is in the stewardship aspect as well, to be able to help with which antibiotic can be paired with which organism more effectively. So there could be some approaches to clinical and diagnostic microbiology as well. But I think the main is to right now is in the early stage of that of the drug discovery process.
Interesting. Okay, lots of questions coming in. We will need to wrap up a little quickly because we have to announce our big giveaway, Anirban. But I think we'll end with one question, last question, which is Raksha's question. When we talk of AMR, we talk about the bug. Largely, we talk about the bug. The bug has this plasmid, the bug has this transposon, the bug has this chromosomal element, and so on. But do we talk enough about the host influencing the antimicrobial resistance development? Like, are there host inflammatory immune factors that could, set up this evolutionary race where if I'm infected with a bug, it's more likely that to be resistant and stick on? Can that happen in one infection cycle, or do you need multiple hosts?
This is a very good question. This gets at, I think, where the future will go. So the first phase of, I think, treating these organisms was, yes, completely focused on the bug. Okay, this is the microbe. It is pathogenic. It's always pathogenic. Let's give it an antibiotic. We don't care about the collateral damage to the body, and we don't care about uniformly treating it as this is what the host response. We don't care about the host response either. So equal dosage, very large amount, basically sort of a nuclear reaction towards that, and not worrying about anything else. Then the second phase was finding out that some of these pathogenic organisms are very context-specific pathogens. So like if it's in the stomach, it's an E. coli. It could be antibiotic resistance, but it's a harmlessly commensal. But then it goes down to the urinary tract, and then it becomes sort of like it's in the wrong place. So then it becomes sort of context dependent. And the question of the host becomes, well, how did it get there? Like, what were the immune responses in the host? What were the inflammatory responses? How did it actually get from one part of the body to the other? But even moving beyond that, now we're sort of at the third stage of where we think about the pathogen as not specifically being sort of in this vacuum, but especially if we're talking about the gut or the other parts of the body that actually have a microbiome. It's interacting with not just the host, but it's also interacting with other bacteria. And I think the key example of this is C. diff, Clostridium difficile or Clostridium difficile, as it was known before. And that is like, you could be more prone to getting this infection if you have sort of this dysbiosis in your own system, and you have certain effects of how your own body is reacting. And that also even ties in with what you're eating and how you're feeding your own microbiome. So there's this whole ecosystem of the pathogenic organism interacting with other organisms, other microbes, and with host cells. Then we are only just scratching that surface. So I think that is going to be a space to look at for the next 5 or 10 years at least.
It's a great question, Anirban, and brilliantly answered by you. So great question, Raksha. Great answer, Anirban, here and in your book, because there's a whole chapter that says the world within us, where you move from the world outside us, which was all prospecting for soil microbes and their chemicals, but within us, it's not only host immune, genetic, and inflammatory factors, but the host commensal microbiome itself. So this is quite literally a whole world to look at within us. So there's a whole chapter on it, Raksha. And if you're a winner of the complimentary giveaway, or you can buy the book. All right. Okay. So we're going to wrap up. And even we tried to condense as much as we could in 1 hour, but I think it was a very engaging discussion. Lots more to talk about. We'll try and keep our collaboration and our discussions going.
Yeah.
It's time to do a few things now. It's time to announce who our secret judge was. Well, the secret judge is our program manager, science communication, Ankita Rathore. Thank you to our secret judge. And secret judge will now announce the complimentary giveaway. Go over to you, Ankita.
Yeah, thanks, Karishma. So, hello everyone, and thank you to Anirban for answering all the questions very patiently, and thank you to our participants for keeping the chat window very active. So we are giving away 4 books, and we have the winners now. Akshata Puri Bajaj. Second would be Ashwini Kumar, Chetna Baliga, and Raksha Kochi. Thank you for all your thought-provoking questions in the chat window. And to receive your books, we would be needing your details. So I would request you, the 4 of you, to reach out to us on hello@indiabioscience.org. And you can send us your details there and we'll ship the books right away. So thank you so much.
That is wonderful. Thank you, Ankita. That was a congratulations to all the winners. I hope you can see my slides, everyone. So winners, where are you? How do you feel? Let me find you in the chat window. Wonderful, great. Everyone seems to be really happy. So do not forget to email us at hello@indiabioscience.org with your details so we can ship you the book. Okay. All right. Okay. So that was that. Thank you, Ankita. And now we'll move on to a little exercise for you. We want to keep these virtual book chats going. We understand that they cater to a niche audience. Maybe that's what we'd call it, right? But the quality of the discussions is very robust. And at India Bioscience, it gives us a chance to put science in the center of India Bioscience, right? So please give us your feedback so that we can try and curate more such sessions for you. So maybe whip out your phones and give us your feedback through this QR code. Okay, all right, we're close to wrapping up. Okay, the link doesn't seem to work. We're not sure why. Arushi, you just want to put the link in the feedback chat window? The feedback link in the chat window? Okay, we will put the link here. Thank you, Suresh. Thank you, Akshita. So as you can see, Anirban, quite a lot of engagement. Oh, Arushi's already put the link. So that's great. Thank you, Arushi. Oh my God, I don't know why this is coming up. Okay. If you can still see my slides, I will move on a little bit. The link to the feedback form is in the chat window. And that's us. So stay engaged. Let's enable change together and get in touch with us on email for any questions, ideas. We look forward to hearing from you. All right. So thank you very much, Anirban. I mean, really, this could not have been possible if you hadn't written the book and if you hadn't been ready to come on board for this session. So quite literally, thank you. And thank you very much to Juggernaut Books, our partner for this session, for the coming on board immediately with just an email. So thanks to Bioma, and also for giving us these complimentary books that we can give away to you.
Yeah, thank
All
you.
right, so
I
thanks so much.
absolutely loved it. It's actually it's 1:30 a.m., but I feel like it feels like middle of the day. So I really enjoyed it. Thank you so much.
Sounds good. Sounds good, Anirban. Thank you so much. Thanks, John. India Bioscience does that to people. We energize people at midnight. You know, we do that. People get excited after our sessions. They are happy. They go back thrilled with new ideas. Of course, you have no dearth of ideas. So we're looking forward to your next books that are going to come along the way. No pressure, but we look forward. And we'll be in touch. Okay.
Absolutely.
Bye-bye, everybody. Have a happy Friday and good weekend. Take care. Bye-bye.
Thank you.
Bye. Bye, Anirban. Take care.